Okay, so in today's activity, this vanilla wafer with icing on it is going to represent a red blood cell. So as forensic scientists, when we get to a crime scene and we find blood, we need to figure out what blood type it is. There are 4 blood types. They are listed here. There's A, B, AB, and O. What makes them different, you ask? Well, the donut-shaped red blood cell is the same in all of these, but what's different is what's on the surface. So these little letters represent what are known as antigens. Antigens exist on the surface of a red blood cell. So to show, for example, what an A blood cell will look like, I'm going to use green M&Ms with the letter M face up to represent antigens on the surface of a red blood cell. Now, you'll get to eat in this lab, as you see. So this is a blood cell. That's an A blood cell. A B blood cell, for example, would look like this. It would have B antigens on the surface. An AB blood cell, as you can guess, would have both A and B antigens on its surface. But what would an O blood cell look like? We don't have O antigens in our body. We can either have A antigens or B antigens. So what, you might ask, is an O blood cell? Guess what? If I have a regular blood cell that I get from my box of blood cells here, and it has no antigens on the surface, nothing sticking up off the surface, that is an O blood cell. And so these are the 4 main blood groups. Now, as you all know, when you get sick, right, and a virus or bacteria enters your body produces antibodies to try to kill that virus or bacteria. The same thing happens with blood. So if you are an A-type blood person, your body will reject B-type blood and produces what are known as B antibodies. So B antibody will be a green M&M face down. This B antibody will attack anything that has an B antibody will attack anything that has a red antigen or a B antigen. The B blood cells have anti-A antigens. These will attack anything that has a B antigen on the surface. A B blood cell has no antigens. Because it has both A and B antigens on the surface, it can accept all. An O blood cell has both anti-B and anti-A antibodies. So let's see what happens when we do a blood typing test. And what that means is we're going to see that blood found at the crime scene, whether it's A, B, AB, or O. So we're going to take some blood. Right? That is B blood. And I'm going to mix it with A blood. So what happens now, the anti-B antibody will attack this blood cell, and what happens is a process called glutination. Normally, the red blood cells would be spaced out. In glutination, if the antibody attacks the blood cells clump together. And so what we'll see is a clumping of the blood cells in the blood. It'll actually look like little clots of blood. And that tells us that we've encountered an antibody. What if I take this A blood and what if I take this B blood and I mix it with more B blood? Nothing will happen. So what we will see is no clotting or no agglutination. The blood will stay together. So what you're going to do today, before we get to the actual lab part, is with vanilla wafers, I want you in each of these boxes to predict whether or not the blood will clot or clump or not. Because as forensic scientists, when we see clumping, we know that we've had an antibody reaction, and we can use that information to tell us what the blood type we have is. That's it.